Hello, ISAP members and fellow students. My name is Benedicta Chinwoke Okeke-Nwolisa, a PhD student of medical microbiology in the Department of Medical Laboratory Science, Faculty of Health Sciences and Technology, College of Health Science, Nnamdi Azikiwe University, Nnewi Campus, Nnewi, Nigeria. I am being supervised by Professor Ifeoma Besi Enwani Nwokelu and Dr. Kingsley Anukam, who introduced me to the concept of microbiome and bacterial metabolic functions. The title of my presentation is Gut Microbiome, Compositions of Watery Diarrhea and the Effects of Fresh Soursop Fruit Juice as a Dietary Complement in the Management of Under 5 Children with Acute Watery Diarrhea in Onitsha, Nigeria. The World Health Organization (WHO) defined diarrhea as having 3 or more loose or liquid stools than normal for any individual. Gastroenteritis is the name given to diarrheal episodes in children, and so in this presentation, acute watery diarrhea and acute gastroenteritis (AGE) are used interchangeably. Acute watery diarrhea or acute gastroenteritis accounts for approximately 1.34 million deaths annually in children younger than 5 years. That's the study population. This study was therefore set to ascertain the effects of fresh Annona muricata fruit juice as a dietary complement in the management of acute watery diarrhea in under 5 children. Materials and methods. A total of 24 stool samples comprising 12 each collected before and after 3-day administration of 90 ml each of fresh Annona muricata fruit juice from 10 under 5 children with acute gastroenteritis and 2 apparently healthy controls. The dosage given was 10 ml times 3 daily per participant. These diarrheic stool samples were collected immediately after bowel movement with a swab stick and then inoculated into ubiome kits prior to 16S metagenomic analysis. The extracted bacterial DNA was sequenced for V4 region of the 16S ribosomal RNA using Illumina MiSeq platform. PyCrust was used to predict the metabolic functions from the 16S ribosomal RNA gene dataset. The bacterial community relative abundance data and in silico predictions for bacterial metabolic functions from gut samples in both pre and post administration of fresh Annona muricata fruit juice from test and control participants were analyzed and compared. Results. On average, there was no significant difference in the alpha indices typified by ACE and Shannon index. The beta diversity showed different clustering positions with principal coordinate analysis. However, at individual taxonomic categories, there was a marginal increase in some genera associated with gut health, such as Bifidobacteria, Faecalibacteria, Enterococcus, and an increase in Firmicutes and bacterial death ratio. On the other hand, some bacterial genera associated with acute gastroenteritis were decreased in their relative abundance after administration of fresh Annona muricata fruit juice. For example, Clostridium and Bacteroides. There was downregulation of some bacterial metabolic functional genes associated with gut dysbiosis after consumption of fresh Annona muricata fruit juice, whereas some bacterial metabolic functional genes were upregulated, such as malate dehydrogenase, complex iron sulfur molybdenenzyme family reductase subunit alpha, polypyrimidine tract binding protein 2, and significantly, xylan alpha glucuronosyl transferase. It is important to note that the gene autolog xylan alpha glucuronosyl transferase was turned on in subjects after Soursop consumption, which may influence the production of short-chain fatty acids such as butyrate and propionate. Non-digestible dietary fiber derived from plant material is highly enriched in the lignocellulosic polysaccharides, cellulose and xylan. Genes encoding enzymes involved in xylan degradation have been identified previously in bacteroidetes family members from the human colon, such as Bacteroides ovetus and Bacteroides xylanisolvens, and some strains of bifidobacteria. Fresh Anona muricata fruit juice was generally well accepted by the under 5 children with acute gastroenteritis and contributed to their better recovery outcomes. Conclusion. Our findings suggest that fresh Anona muricata fruit juice taken orally increases the relative abundance of some healthy bacterial communities and decreases the relative abundance of some bacteria associated with gut dysbiosis in acute gastroenteritis. In addition, there was putative upregulation of genes for degradation of non-digestible fibers. Acknowledgement. I wish to acknowledge sincerely all the parents of the participants who voluntarily consented to this study. I equally wish to acknowledge my clinical collaborators, Professor Ifeoma Ebuonu and Dr. Amala O. Odita, who helped in the development of the clinical aspects of the study instrument and assessment and monitoring of the participants throughout the course of the study. Most importantly, I wish to express my sincere gratitude to Dr. Kingsley Anukam for funding this study, which was made possible under an academic consortium grant in kind awarded to him by microbiome in 2018. Thanks for listening.